Hello dear students. In this lecture we are going to see the point called physical mutagenic agents, which include UV and X-rays. Mutagens, these are the agents which are responsible to cause the mutation. Then these agents, it may be physical type or it may be chemical type. Okay, so first, we will discuss about the physical mutagenic agent. So, in this lecture, we are going to focus mainly on UV and X-rays. Okay. And plus, we are going to see about the base analog. What are the base analog and what are the example? Let's begin it. Radiation, it was the first mutagenic agent to be known in the year 1920s. Okay, so what this radiation do to the DNA? Let's see it. Radiations, it damages the DNA, but how it damages the DNA? By breaking the double strand break. Okay, it may break the double strand and create the gap between this double strand, or it may lose the base. Okay, any base it may lose or it may remove, or it can change the base. Instead of, we know that adenine, it base pairs with thymine, so instead of this normal base pairing, it may add the another base pair which may create the mutation. So it may lead to the base change, or it may cause the cross linking. Okay, so like that, adjacent cross linking, or we can say instead of complementary base pairing, there will be formation of this cross linking, or it may break the single strand. It may cause the formation of the dimer. Okay, dimer means instead of complementary base pairing, there is adjacent base pairing. Okay, which leads to the formation of the dimer. Usually there is formation of the thymine dimer. Okay, because there is adjacent base pairing between the thymine nucleotide. Or it may cause the breakage of hydrogen bond. This hydrogen bond is present between the nucleotides. Adenine is base pair with the thymine with 2 hydrogen bond, and guanine is base pair with cytosine with 3 hydrogen bonds. Okay, so this radiation, it may break this hydrogen bonds. Now there are two types of radiation. One is ionizing radiation and second is non-ionizing radiation. What is ionizing radiation? These are the electromagnetic waves that have sufficient energy to ionize the atom and detach the electrons from that atom. That is known as ionizing radiation. Okay, and well-known example of ionizing radiation is X-rays. Okay, so what are ionizing radiation? These are the electromagnetic waves which have lot of energy to ionize the atom, so it forms the ions. Okay, so that is the ionizing radiations. There are different examples of radiations like X-rays, gamma rays, cosmic rays. Okay, this all the rays are capable of penetrating the tissue and damaging the DNA. Okay, so it can damage the DNA by breaking the hydrogen bond, by breaking the strand of DNA, by formation of the dimer, etc. Okay, so what is the mode of action? The mode of action is when radiation fall. Okay, so what they do, they remove the electrons from the atoms, changing the stable molecule into free radical and reactive ions, which ultimately change the structure of base and break it. Okay, so that is the mode of action. Okay, क्या करता है जब radiations fall होते हैं, तब ये electrons remove करता है from the atom. Okay, and it forms the free radical and reactive ions. It creates the formation of radicals and reactive ions. As there is formation of free radicals and reactive ions, it can change the structure of base, or it can break the phosphodiester bond present in the DNA. Okay, phosphodiester bond, that is nothing but the sugar and phosphate backbone. Okay, so it can also break the sugar phosphate backbone. Ionizing radiation, this causes the formation of ion, as we have seen just now. Okay, what it does, it causes the formation of ion. This ion react with the nucleotide, and even they can react with the sugar phosphate backbone, and we call it as mutation. This mutation can be repaired by the repairing system called as nucleotide excision repair. So this radiations have been fall onto this tissue. Okay, so it penetrates onto this tissue, and the gene of this tissue get break. See, by the radiation there is breakage of this double stranded DNA, and as there is breakage of this double stranded DNA, the nucleotides have also been separated from this strand, and ultimately it leads to the generation of cancerous cell. Okay, or it may lead to the disease called as cancer, growth of the tumor, unwanted growth of the cells. Okay, so it leads to the cancer. So radiations, it may cause the cancer. If we will see the how actually this UV, it creates the mutation. What is the mechanism behind the mutation? So when UV radiations are fall onto this gene, okay, onto this normal gene, when UV radiations are being bombarded, it leads to the formation of thymine dimer. Okay, means in this case what happened, instead of complementary base pairing, instead of base pairing of one strand of nucleotide to the other strand of the nucleotide, there is base pairing between adjacent nucleotide of single strand. ठीक है, जो adjacent होते हैं उनके बीच में pairing होती है, and that pairing occurs to this thymine nucleotide, which leads to the formation of thymine dimer, or you can simply say thymine dimer, because dimer itself stands for the two. Okay, so this mutation, it may be repaired by using different enzymes. Actually, repairing is not our aim. Our aim of this lecture is how the mutation is been caused by using the radiations. Okay, by using different radiating agents. So by using the UV, there is formation of thymine dimer. And how the dimer is being formed? The dimer is being formed by the adjacent base pairing instead of complementary base pairing. So we call it as a pyrimidine dimer also. So this is the formation of the thymine dimer. Okay, so instead of complementary base pairing, there is a adjacent base pairing. Okay, so like this, onto the one strand thymine base pair, instead of this adenine and thymine base pairing. Okay, so this is about the formation of the dimer. Now how, with the help of the X-rays, the mutation is been caused? This X-rays, it produce the reactive ions. Okay, because it is the ionizing radiation, and ionizing radiation have very much large energy to ionize the atoms. Okay, and to produce the reactive ions. So this X-rays, what it does, it damages the base and sugar residues. Okay, it damages the base. This is the base. Okay, so it damages the base. When X-rays are fall, it damages the base, plus it disturbs the sugar phosphate backbone, or it damages the phosphodiester bond. Okay, so that is very harmful. So that they are of two types, alpha particles and beta particles. Alpha particles, they have two protons and two neutrons. Beta, they have the electrons, and this alpha and beta, they have the ability to penetrate inside this nucleotide. They disrupt they damage the base pairs, and plus they disrupt this sugar phosphate backbone. So that was about the physical agents. In that, radiation we have discussed, and in radiation specifically we have discussed about the UV, which causes formation of thymine dimer, and X-rays, which leads to the disturbance in bases, and also it damages the sugar phosphate backbone. Okay, so that was about the physical mutagens. Now let's focus on to the chemical mutagens. Chemical mutagens, as I told, in this lecture we are going to see base analog. Okay, so chemical mutagen was made by the scientist called Charlotte Auerbach, and the base analog is the very major and important mutagen under the chemical mutagen, because it uses the carcinogenic chemicals. Okay, so chemical, what are the base analog? Let's understand the definition of this base analog. Base analog, these are the this base analog, it can mimic your normal nucleotide bases. Okay, or it can copy the normal nucleotide bases. That is the base analog. Base means your nucleotide bases. It may be any cytosine, thymine, uracil, adenine, any. Okay, so these are the bases. And analog means same दिखने वाले, उसको मिमिक करते हैं, उसकी कॉपी करते हैं. Okay, that is the meaning of this word base analog. Chemicals with structure similar to that of any of the 4 standard bases of DNA. So these are the analogues whose structure is similar. Okay, so we can say that these base analogues, they have the same molecular structure. As I told that it can mimic or it can copy. Okay, so these are the chemicals with the same structure. DNA polymerase cannot distinguish these analogues. They may be inserted into newly synthesized DNA molecule. So these are base analogues, they have the same molecular structure as that of our normal nucleotides. So our enzyme DNA polymerase, which is used for the replication purpose, this enzyme, it cannot identify the difference between original analog and duplicate analog. Okay, so what it does, it polymerizes. Okay, and there will be formation of the mutated gene. So we will see two examples of base analog. One is 5-bromouracil and second is 2-amino purine. These are the structure of your normal bases: adenine, guanine, thymine and cytosine. So let's first focus on to this bromouracil, which is also called as 5-bromouracil. Why 5? Because at the 5th position, or you can say, what is the difference between this bromouracil and your normal nucleotide? Let's understand this. So base analogues, what are this? These are we know that they have the same molecular structure. So bromouracil, it is artificially created compound used in the research purpose. तो अभी किसके जैसे मिमिक करता है, किसके जैसे कॉपी करता है? It resembles like thymine. Okay, this bromouracil, it resembles like thymine. It has the bromide atom instead of methyl group and will be incorporated into DNA, and it can pair with the adenine like thymine. Okay, so this bromouracil, it can mimic like thymine. Okay, mostly it mimics like a thymine, and in rare cases it mimics like cytosine also. Okay, so that you have to keep in mind that bromouracil, it mimics like thymine, and we know that thymine base pairs with adenine. So this bromouracil, ultimately it is going to base pair with the adenine, same as that of the thymine. It has a higher likelihood for tautomerization to the enol form. Now there are two forms of this 5-bromouracil. So this is the structure of 5-bromouracil and this is the structure of normal thymine. This at the fifth position, okay, at the fifth position, instead of methyl group there is bromide group. Okay, there is bromide group, hence the name 5-bromouracil. So आप देख सकते हैं स्ट्रक्चर सिमिलर है, सिर्फ फिफ्थ पोजीशन में मिथिल ग्रुप की जगह यहाँ पर ब्रोमाइड ग्रुप है. Okay, so it is called as the 5-bromouracil and it exists in two form. Okay, बहुत ही dangerous है, दो फॉर्म में ये एग्जिस्ट करता है. One is keto form and second is enol form. Okay, keto form, it consists of double bond O group. Okay, keto bond, it consists of double bond O group, and enol form, it consists of hydroxyl group. Now keto form is the form in which it is present at the, or it is present like thymine. Okay, so in the keto form, this 5-bromouracil, it acts as a thymine, which is very common, and in the enol form, this 5-bromouracil, it acts as a cytosine, which is rare. Okay, ये cytosine क्या है? It is the rare form, और thymine क्या है? It is the common form. So that has been there. And when this 5-bromouracil, it behaves like a thymine, it can pair with the adenine, and at the enol form it behaves like cytosine. So when it is in the cytosine, it can pair with the guanine. Okay, that you have to keep in mind, the difference between these things. Okay, so yes, let's see the mechanism. The mechanism is just keep in mind the thing that 5-bromouracil under normal condition or commonly it behaves as a thymine. Okay, so commonly 5-bromouracil, it behave as a thymine, and it base pairs with the adenine. Okay, and in rare condition, this 5-bromouracil, it base pairs with the cytosine. This is the rare condition and this is the common condition. Okay, so that thing you have to keep in mind. So yes, let's see the mechanism. Suppose this is the gene. Okay, that gene consists of a double stranded DNA is there. It consists of thymine and adenine sequence. Now when there is mutation by this agent called as 5-bromouracil, what it does, we know that this 5-bromouracil, it mimics to the thymine. Okay, it can mimic the thymine. So instead of thymine, there is addition of this 5-bromouracil. Why? Because this 5-bromouracil, it can mimic like thymine. So here instead of thymine, there is 5-bromouracil and adenine is there. After replication, this is the first replication. After second replication, instead of, now this is the common form. Okay, this is the common form. Now in the second replication, there is a addition of rare form, and rare form is the, we know that rare form is cytosine. Okay, so here 5-bromouracil, here instead of 5-bromouracil, we know that cytosine is there. Okay, and here 5-bromouracil means your thymine is there, which is the common form, and this is the rare form. Okay, so here पहले common form attach हुआ है first replication में thymine और second replication में cytosine attach हुआ है, और cytosine के complementary guanine attach होगा. Okay, and during the third replication during the third replication, there will be formation of the cytosine and guanine. Okay, so this is the mimic form. We want to remove this 5-bromouracil. It has been removed and just cytosine is been there. So we get from AT to the GC. So this is the conversion of AT to GC, that is called as the transition. Okay, transition means it is a purine is replaced by purine and pyrimidine is replaced by pyrimidine. Okay, so that is called as the transition. So your T is converted into C and A is converted into G. So that is called as the transition. It is very simple. We have the normal gene, that is TA. Okay, 5-bromouracil is been added. So 5-bromouracil, it acts as a thymine. So to the thymine, the complementary base pair adenine will get attached. Now at the second replication, the rare form called as cytosine will get attached, and to the cytosine, guanine will get bind. And we want to remove this 5-bromouracil because it is the not original cytosine. Okay, it is the duplicate cytosine. Then next it has been removed and just your cytosine and guanine has been there. Okay, so that thing you have to keep in mind, and it is called as the transition type of mutation. Okay, even how this AT is been converted into GC, in the same way GC can be converted into AT. Okay, so this is the GC. This is the GC, and we know that cytosine is the rare form of 5-bromouracil. So instead of cytosine, there will be addition of 5-bromouracil in the first replication. During the second replication, what will happen? So here 5-bromouracil is nothing but it is your cytosine. Okay, so it is your cytosine, and after the second replication, here it is the rare type, and in the second replication, common type of 5-bromouracil will get bind, and that is your thymine. Okay, so to the thymine, adenine will get bind, and to the cytosine, guanine will get bind. And we know that this is the duplicate form. So we remove this 5-bromouracil, and there is addition of just thymine and adenine. Okay, so finally there is from GC there is conversion of AT. So this is very simple conversion, how this is the replication process, how actually it takes place from AT to GC. So this is the AT gene replication 1. In the replication 1, you will see that first, instead of thymine, there is 5-bromouracil. Okay, these two strands get separated. So instead of thymine, there will be addition of 5-bromouracil, and this parental strand will be same. Okay, this is the daughter strand, this is the parental strand. So during the second replication, AT is the normal, so we will leave that, and the rare form will get attached, that is cytosine. Okay, so to the cytosine, guanine will attach. At the third replication from AT, there will be generation of GC. Okay, so that is very simple how from AT to GC conversion is there, and from GC to AT conversion is there. Second is 2-amino purine. Now 2-amino purine, same mechanism is there, but difference is in the analog. This 2-amino purine is the analog of adenine. Okay, this 2-amino purine, it behaves like a adenine. So this adenine, it can pair with the thymine. So we can say that common form is the adenine. Okay, and rare form is the cytosine. Okay, that you have to keep in mind. Common form, normal condition, it behaves as an adenine, and in the rare condition, it behaves as a cytosine. So let me write that. Okay, so in the normal condition or in the common condition, it can behave as a adenine, which will base pairs with the thymine, and in the rare condition, it can behave as a cytosine. Okay, which will base pair with the guanine. This will help you to remember the things. Okay, so this is the normal gene TA. when there is addition of 2-amino purine. Okay, so in the common form 2-amino purine is the adenine. Okay, so in place of adenine our 2-amino purine is been added. Okay, this is the common form. And at the second replication there is addition of the rare type, and rare type is, we know that rare type is cytosine. Okay, so here cytosine is been attached, cytosine or guanine. So we know that complementary is there. So here to the cytosine guanine is been attached, we have to remove this 2-amino purine because we know that it is mimic form. Okay, so it has been replaced by guanine. Okay, so from AT there is conversion of GC. This is the simple mechanism of 2-amino purine. Same you can convert from CG to the AT. So this was all about the base analog. Hope it will be clear to you. Thank you.